in collaboration with Genetic Engineering and Biotechnology News. The webinar is entitled Vanquishing the Virus, the Top COVID-19 Drug and Vaccine Candidates. Thank you so much for joining us this afternoon. My name is Nancy Kelly, and I serve on the steering committee and as a founding member of NYC Builds Bio. I want to thank Alex Philippidis, Senior News Editor, and Kevin Davies, Executive Editor at GEN, for doing the hard work of scouting out and assembling all of the information you'll learn about today. I salute the New York City companies and institutions that are pursuing these treatments, and I would also like to acknowledge the founding and corporate members of NYC Builds Bio who make this programming possible. This is the second of 4 virtual events hosted by NYC Builds Bio this month. If you haven't already, please be sure to check out and register for the programs on the NYC Builds Bio website. Last week, we hosted NYC Builds Bio first virtual quarterly breakfast, attended by over 100 registrants, which focused on the heroic efforts made by companies, institutions, and many of NYC Builds Bio members to produce and expand the testing capacity, personal protective equipment, and facilities needed to care for patients during the COVID-19 crisis. You can access the event presentation and recording on the NYC Builds Bio events page on the website. On May 21st, in collaboration with BioCentury, we are hosting a networking reception for current and prospective members, which will include a discussion surrounding the business and financial impact of COVID-19 on the life sciences industry and healthy building employee strategies for returning to work. Bill Harvey of Newmark Knight Frank will also give a virtual tour of InnoLab's Long Island City King Street properties, which is currently under construction here in New York City. Over 200 people have registered for this, and we will allow time for member breakouts at the end of the call. On May 28th, SGA will host the first program of our newly launched virtual seminar series, also in collaboration with Genetic Engineering and Biotechnology News, entitled Life Science Facility Planning Post-COVID-19. The program this afternoon provides an in-depth look at the vaccines and treatments for COVID-19 under development. As the threat of the novel coronavirus continues to grow globally, innovative pharmaceutical companies and academic institutions are ramping up efforts to study the virus and develop vaccines and antivirals. The development pipeline for COVID-19 now includes more than 200 candidate drug treatments and vaccines, 8 of which are already being treated in humans. We will hear from Alex Philippidis, Senior News Editor at GEN, and the executives from 3 New York companies and institutions that are actively working on vanquishing the virus: REGENERON Pharmaceuticals, Tonix Pharmaceuticals, and Mount Sinai Innovation Partners. But before we begin, I just wanted to cover a few administrative details. There's a program and PowerPoint highlighting the hosts, sponsors, speakers, and projects for the meeting. These will be posted on the NYC Builds Bio website after the meeting. The program is being recorded and will be available on the website and the NYC Builds Bio YouTube channel after the meeting. If anyone's having technical difficulties accessing the Zoom room or other technical issues, please email me at info@nycbuildsbio.org or send a notice through the chat button at the bottom of your screen. Since most of you are participating in listen-only mode, please feel free to submit your questions via the Q&A button at the bottom of your screen. We will open up for discussion and Q&A at the end of the program, and will address as many of the audience questions as we possibly can. Now I will open it up to Alex Philippidis, who will begin the discussion.
Okay, thanks, Nancy, for your kind introduction, and thanks for inviting me to speak today at the webinar. It's fitting that this webinar is titled Vanquishing the Virus, And it's so because it's what everyone who's lived through these difficult few months yearns to see happen, and certainly what Jen would love to report someday and soon. Which brings me to a word from Jen, which is co-sponsoring this webinar. Next slide. Genetic Engineering and Biotechnology News was launched in 1981 by Marianne Liebert herself as the first publication devoted exclusively to biotech. Our key reader segments are R&D researchers, lab managers, bioprocess development scientists, and executive management. Our reporting focuses on 6 key pillars: bioprocessing, cancer, omics, translational medicine, genome editing, and drug discovery, which is my focus as it relates to COVID-19. COVID-19 is caused by a sphere-shaped virus called SARS-CoV-2. Now, what's in a name? SARS stands for Severe Acute Respiratory Syndrome. CoV is short for coronavirus. The numeral 2 means that the virus is a genetically related second version of the coronavirus responsible for the SARS outbreak of 2003 called SARS-CoV. You may be seeing the figures from Sunday on the slide in terms of cases and deaths, but today the numbers are worse. As of today, total deaths from COVID-19 reached more than 319,000 worldwide, about 30% of them in the United States alone, just over 90,000 deaths. The number of confirmed cases stood at more than 4.8 million as of today, nearly 1/3 of those also in the US. Now, during a webinar by Chemical and Engineering News earlier this month, Derek Lowe, who's a longtime observer of the biopharma industry through his In the Pipeline blog in Science Translational Medicine, offered a sobering view of how hard he expects drug development to be versus COVID-19. Quote, "We're not going to have a silver small molecule bullet." Well, that has not stopped hundreds of drug and vaccine developers, as well as academic researchers and their institutions, from pursuing therapeutics against COVID-19. As of Sunday, no fewer than 216 drugs and vaccines tracked by Jen were in development against the virus, including the 8 vaccines that have advanced to clinical trials. Among treatments categorized by Jen are those of the 3 developers we'll be hearing from today. REGENERON generated buzz a few days back for its antibody cocktail, which is set to start clinical trials next month. I'll talk a little bit about that more later. Regeneron's also part, I'm sorry?
Can you
Regeneron's
just
also part,
talk about, could you advance the slide to where it's supposed to be? Or just tell Carolina where to advance the slide.
Yeah, yeah, that needs to be further advanced. Here we go. Past that. One more. One more, please. All right. Here we go. As of May 17th, 217 drugs and vaccines in development. And I should point out that this week, Jen is going live with a revised version of its list. It's more of a tracker, or allowing for updates as news developments happen. And you'll see access to that on our website, genengnews.com, when we're ready to come out. And that should be in the next day or two.
Alex, we've got great representation from the New York companies today. In addition to those highlighted in the webinar, are there other New York companies we should know about?
Oh, yes, absolutely. There's a whole bunch. Here's some good examples of that. Pfizer, which is headquartered in Manhattan, is a co-developer with BioNTech on BNT162, the mRNA vaccine. It's begun clinical trials. Montefiore Medical Center, Albert Einstein College Medicine, is a partner in a convalescent plasma clinical trial by a consortium that also includes Johns Hopkins and the Mayo Clinic and Takeda. Applied Therapeutics out of New York is conducting trials of AT-001. It's a lung inflammation and cardiomyopathy drug, but they're testing it on COVID-19 patients at several hospitals in the city, including Mount Sinai. Ho Therapeutics, also based in New York, is working with Voltron Therapeutics and Mass General's Vaccine and Immunotherapy Center to develop a COVID-19 vaccine. And we've also seen a lot of trials take place in hospitals in New York because of the large number of cases in our area.
Great, Thank you.
Right. So looking at types, one of the points I wanted to make is that most, though not all, of the therapeutics in development fall into four categories. Antibody therapies bind to a specific antigen in order to destroy it. Now, antibodies are blood proteins produced in response to an antigen, a virus or other foreign substance that triggers immune response. Antivirals inhibit but do not destroy their target pathogen. Broad-spectrum antivirals target multiple pathogens. Then RNA-based therapies use messenger RNA strands that code for a disease-specific antigen. Once the mRNA strand is inside the body's cells, the cells use the genetic info to produce the antigen. That antigen is then displayed on the cell surface, where it is recognized by the immune system and destroyed. Then there are vaccines, which prevent disease by helping the immune system recognize and fight pathogens that include viruses, such as SARS-CoV-2. Next slide, please. Jen assigns COVID-19 drugs and vaccines to any of four categories. The front runners are categories that have become leading candidates based on public attention or advanced stages of clinical activity or positive data or all of the above. Definitely maybes are candidates in earlier clinical phases with promising partners, plus more advanced candidates that have generated more uneven data. Are keeping an eye on candidates show promise based on innovative technology or developers with past success or both. And our too soon to tell candidates appear to be long shots pending additional details from their developers and or clinical progress. Next slide. I'll run down 10 of the front-running therapeutic candidates, 5 drugs and 5 vaccines, and what we should look for from them in the coming months. Among drug candidates, next slide, I'll start with Regeneron's antibody cocktail. The company's developing a lead combination of neutralizing monoclonal antibodies using its VelocImmune mouse platform. A clinical trial is expected to begin in June. Company's aiming for a couple of 100,000 doses to be made by summer, ultimately up to 1 million doses a month. And should the lead candidate not work out, Regeneron also has a backup cocktail in development as well. Next slide. Hydroxychloroquine and azithromycin. Boy, have we heard those in the news. And it was sort of rekindled the whole controversy yesterday with President Trump's comments, which we all saw and heard. Yet Washington has actually sent a mix of signals. The FDA in March issued emergency use authorization, allowing for healthcare providers to make hydroxychloroquine or chloroquine available for patients for whom a clinical trial was not available. And then a month later, at the end of April, the FDA issued an advisory to the healthcare professionals cautioning against the use of hydroxychloroquine or chloroquine due to the risk of heart rhythm problems potentially. And we are seeing, however, some phase 3 trials in the works, one by Novartis, which has a generic version of hydroxychloroquine. One also announced last week by the NIH, and that's a phase 3 trial that is set to, it actually has started, they had reported. Next slide, please. Remdesivir is an antiviral drug that was granted emergency use authorization by the FDA at the start of the month, following what were promising interim phase 1 results showing a median time to recovery of 11 days for Remdesivir versus 15 for placebo. Anthony Fauci, the head of NIAID, we've all seen in recent weeks, had called it a very important proof of concept, but last week he sort of dialed it back a little and said it was statistically significant, but also really modest. Now, not all the trials of Remdesivir succeeded. A phase 3 trial in China did not, in 158 patients with severe version of COVID-19. The NIAID trial is for patients with a mix of symptoms. However, Gilead is talking about ramping up the manufacturing to 500,000 treatment courses by October and up to 1 million by the end of the year. So next slide. Two other drug candidates worth watching. Distributed Bio, it's a company that bioengineers broadly neutralizing antibodies based on its superhuman platform. They're now testing antibodies on hamsters, they told the San Diego TV station last week. Based on hundreds of potential candidates that were seen to block ACE2 receptor interaction with the virus. And that fellow in the photo is CEO Jacob Glanville. He's a PhD. He was featured in January by Netflix in the documentary Pandemic, How to Prevent an Outbreak. Avigan, which is favipiravir, made by Fujifilm's Toyama Chemical subsidiary, is also worth watching. Back in March, the drug had generated positive clinical trial data in China, including a shortening of the time after treatment that for patients to test negative for COVID-19. From 11 days without treatment to 4 days with Avigan. And as a result, Prime Minister Shinzo Abe of Japan had said his country will stockpile 2 million treatment courses, and Fuji's talked about increasing its manufacturing capacity for the drug. Next slide. Starting on vaccines, BNT 162, which I mentioned a moment ago, BioNTech and Pfizer are the developers. If the vaccine works, BioNTech stands to gain up to $748 million from Pfizer. The vaccine's in a phase 1-2 trial, ultimately 7,600 participants, and those include 360 in the first stage dose level escalation in the United States. The first clinical data is expected at the end of June or in July. Next slide. Chadox-1, which is the University of Oxford, its spin-out company Vaccitech and AstraZeneca. Now, AstraZeneca joined the effort at the end of April by saying they'll oversee global development, manufacturing, and distribution of the vaccine candidate. It is in a phase 1-2 trial now, up to 1,100 healthy volunteers. And AstraZeneca has agreed to provide the UK with access to the vaccine as soon as possible, should it succeed in clinical trials. And in a separate study that was announced last week by the NIH, the vaccine appeared to work in 6 monkeys by protecting them from pneumonia caused by the virus. Next slide is Moderna's mRNA 1273, and that made a lot of news yesterday because Moderna announced positive interim phase 1 data. All 45 participants across the study's 3 dose levels produced antibodies of the virus by day 15 after treatment. And 8 participants in 2 of the dose levels reached or exceeded the measure of antibodies generally seen in people who have recovered from COVID-19. That's on track for advanced phase 2 and phase 3 trials, with the phase 3 trial set to start in July. And through BARDA, the Biomedical Advanced Research and Development Authority, our government is committed up to $483 million toward development, of course, tied to the vaccine working. Also worth watching, next slide please, is Novavax's NVX-CoV2373, and that's because a few days ago, the Coalition for Epidemic Preparedness Innovations, or CEPI, actually raised its commitment by adding $384 million to a $4 million stake they took back in March. So now CEPI has $388 million invested toward clinical development and large-scale manufacturing of the vaccine. A phase 1-2 trial for that vaccine is planned, set to start this month, 131 patients in the phase 1 portion. Also, CanSino Biologics, which is the developer of China's most advanced vaccine through clinical trials, that's AD5 and COV. They're partnering with a branch of China's military, the Academy of Military Medical Sciences, Institute of Biotechnology, for what development of the vaccine. It's in a phase 2 trial. 508 people were tested as of May 4th, but results are expected as soon as late this month. And it was the first of the vaccine candidates to advance to clinical trials in China. As we look ahead, what we can anticipate, next slide, I see three things. One is speed. The timeframes for developing new drugs and vaccines will continue to shorten because companies are chasing market opportunity and applying technologies like faster sequencing, while regulators are eager enough for therapeutic solutions that they'll accommodate the companies. But there's a challenge, actually a couple, and that's getting the vaccines onto the market, getting access. Stakeholders will have to negotiate pricing with both public and private payers for these, and nations in many cases have extracted promises to receive the first dosage of new drugs and vaccines whose development they have funded. The UK is getting the Oxford vaccine. China will be getting first dibs at its own developer's vaccine. The CEO of Sanofi last week said the US would have first dibs on their vaccine being co-developed with Glaxo because a company Sanofi later acquired received $30 million in funding from BARDA. And although Sanofi later sort of clarified and even backpedaled on that a little bit, I also see a revival of antiviral R&D because you notice a lot of the leaders in COVID-19 therapy development have applied R&D that they had gathered from trying to develop Ebola drugs. And I see other developers following that same playbook as they look to future pandemics. And that's my presentation. Thank you for listening. Let's stay in touch. Thanks.
So Alex, your research identified 200 programs. What are the overarching trends that you're seeing? What countries or regions are really spearheading new drug and vaccine development right now?
Well, I noticed that it's sad that the hotbeds of the virus, China and the U.S., are also the countries that have the most activity going in companies and institutions developing therapeutics. Of those 8 vaccine candidates, they're in clinical trials. 4 of them are from China. And sorry, there is, however, some European presence. BioNTech is based out of Germany. Obviously, the University of Oxford's in England. And I expect that's going to improve because the European Union recently increased the amount of funding for their Innovative Medicines Initiative COVID-19 R&D program from 45 million euros, that's about $50 million, to 72 million euros, that's $78.5 million. So I'm expecting to see a little bit more activity there. Also CureVac out of Germany, another example of a developer there. So yeah, that's how Australia having some Mesoblast, which tested its remestemcel stem cell therapy at Mount Sinai, and CSL Behring, which is a key player in a consortium focused on convalescent plasma. There you go.
Great. David?
Good afternoon. Is the video working? Hold on. There we go.
Coming up.
Here you are.
Good afternoon. So I'm Dave Weinrich. I run the clinical development organization and I co-head all of development for Regeneron. It's a pleasure to be here today. Next slide. So Regeneron is built upon a set of fundamental research technologies that we've been investing in for the last 3 decades. They all start with Velasa something. And this major investment to speed up and also simplify how research is done has led to 7 approved, FDA-approved medicines and a number of new ones that are currently under review. It's this suite of technologies that we have deployed in a so-called rapid response platform against a number of viral pathogens, including the MERS coronavirus, Ebola virus, and are now deploying against SARS-CoV-2. Go to the next slide. What underpins all of this is the ability to go from immunization against a target through screening and to generate initial cell lines extremely rapidly. We've essentially taken a scientific problem and turned it into an engineering one. And as I'll show you on my last slide, we deployed this against the spike protein for coronavirus and generated thousands of potential antibodies, which we could then screen for characteristics that we liked in a matter of weeks. Now, obviously, we can't do this for every single program on this timeline, but the flexibility of this platform means when a pandemic hits or other public health crisis, we can very rapidly develop antibodies to attempt to neutralize the virus. Next slide. And we've already done this before. So we did this against MERS. In that particular case, we went as far as completing the phase 1 study, and then the virus sort of petered out, and we have it still sitting in the freezer, but have not taken it any farther. But this same platform was also used successfully against Ebola virus. So in this case, we developed a 3-antibody cocktail that targets Ebola, and in partnership with the World Health Organization and Doctors Without Borders and the NIH, deployed Regeneron EB3 into the randomized efficacy trial that was being conducted in the Democratic Republic of the Congo. In that trial, Regeneron EB3 was the arm of the study that caused the Independent Safety Monitoring Board to halt the trial for demonstration of superiority and survival against what was the control arm, the standard of care therapy at the time, which was ZMapp. And we are currently in regulatory discussions with the agency regarding Regeneron EB3. So we have some experience that this approach can work, and we've deployed that same platform to develop a cocktail of 2 antibodies against SARS-CoV-2. I'll step away from this slide for one quick second. It was something that was on Alex's slide. If you think broadly about how to beat this virus, it falls into a couple of buckets. And while we're talking about sort of bucket 2, which are therapeutics that could potentially treat the disease or prevent patients from developing the disease as a bridge to a formal vaccine, something we call passive vaccination with our antibody, the first step was really taking off-the-shelf drugs and trying them. And we've obviously released some data around anti-IL-6 receptor, sarilumab, also known as Kevzara. And I think there's an important story to be told that, for those of you who know that story, there was some initial data that came out of China, a single-arm study on the use of tocilizumab, which is another IL-6 receptor blocker by another company, and it looked very, very promising. On the basis of seeing that public data, Regeneron, as well as Roche, who makes tocilizumab, had undertaken randomized clinical trials with a placebo to try and understand whether IL-6 blockade would benefit patients who are hospitalized and have hypoxia, lung inflammation, causing them to have difficulty breathing due to coronavirus. That step of running randomized controlled clinical studies is absolutely critical, because single-arm studies can very easily mislead. And as we had publicly announced when we read out our phase 2 data, in some patients who were receiving IL-6 blockers, patients who had relatively minor oxygen requirements, it did not appear that there was activity benefit from administering IL-6 receptor blockers. That phase 3 for critically ill patients is still ongoing. But that's sort of tier 1, and what we're talking about is right now is tier 2, and then tier 3 would be formal vaccines, kind of like what you get when you're growing up and you go to the pediatrician and get injections. Can you go to the next slide? So when you put all these technologies together, what happens? You get to compress a timeline that can sometimes take from I have an idea to GMP grade material to be able to put into the clinic that can take 18 months, maybe even a little longer, you can collapse that to less than 6 months. Now, obviously, it moves other things out of the portfolio, but these are unprecedented times where all companies that have a technology, that have a possible therapeutic, need to step up the plate and focus on saving the world more than building out their own portfolio. And that's exactly what Regeneron did.
So David, once the vaccine is developed, how will manufacturing be scaled up and how long will that take? And will you do it regionally around the world or here in the United States?
So as far as our antibody, we have a fair amount of manufacturing capability in-house to Regeneron. And as Len has already disclosed, we're conducting scale up at risk. One of the other sort of research investments that we made very early on is in our cell line technology. And it means that each of our cell lines has a high degree of homology with every other cell line that Regeneron has ever created. Again, it's almost an engineering problem more than an individual scientific problem, scaling up each cell line one by one. Consequently, we can get to large-scale commercial manufacturing incredibly quickly. And in this particular case, even before we've put the first antibody into the clinic, we've already started that process to have commercial scale. Now, that's only part of the answer because no one's got enough manufacturing capability by themselves to be able to do this. And I think this is going to require collaboration amongst companies. If something works, how do we band together to take that which works and be able to create enough doses? And that means putting aside certain competitive predispositions that exist when we're competing in other areas, and instead go, well, we've got a bigger task here of trying to save the world, not dominate the market. And without getting into the details, those conversations are already ongoing between Regeneron and collaborators if we show that this works, or maybe when we show that this works. I'll be a little more positive.
Is there precedent for that level of collaboration?
That's a good question. I don't exactly know the answer, but we're certainly giving it a try, and we've had great reception from a number of different places to be able to do it. But it's still early days. I'm hopeful. It's necessary.
Yes.
Next slide. So I'll end here. I don't want to take up too much time, but just to give an idea of how fast we've gone. In January, we began the discovery effort around targeting coronavirus. By March, we were already screening 1,000 potential cell lines for whether or not the antibodies they produced had the right characteristics, where they bound, neutralized, didn't compete against a second antibody so that you could produce a cocktail, which generally we believe is a better approach. It prevents a single mutation in the virus from rendering the virus immune to the therapeutic. We've been having ongoing discussions with the regulatory authority while we're doing scale up, and we are at this moment still on track to get this into the clinic in June. And as I said in answering Nancy's question, we're conducting scale up at risk so that we could have hundreds of thousands of doses by the end of the summer and millions of doses later, even before we've put this into clinical testing to know whether or not it is going to work. But that's the kind of response that's necessary to beat this. So I'll stop there. At the end of this, I'm happy to take any other questions, but thank you for your attention.
Thank you, David. Seth?
Yes, Nancy, thank you very much for having me. Should I begin or?
Yes, absolutely.
Okay, great. Okay, thank you very much. Well, it's wonderful to be part of this program and see the exciting work being done um, by Regeneron and the other companies. This is truly an unprecedented, at least in my lifetime, challenge. So I'd like to tell you a little bit about what we're doing at Tonix. So the next slide, please. We're a public company, so I'll be making forward-looking statements, and I refer you to our regulatory documents for full information about our company. Next slide. At Tonix, we're working on a potential vaccine for COVID-19, and it's based on a vector platform called horsepox. And the candidate that we're advancing, we call TNX 1800, and it's a modified horsepox virus. It's in the preclinical stage, and it is designed to express the spike protein from CoV-2 that was mentioned just recently. So the next slide shows you, this is a schematic of CoV-2 showing there are a number of different potential targets on the virus. But we, like many other companies, have focused on the spike proteins. These are the proteins sticking out of the surface that are involved in binding to the cells that it infects. And our approach on the next slide has been to clone the spike protein into our horsepox vector system. And at the top is an unmodified horsepox. The blue line is its normal chromosome, about 200,000 base pairs. And the strategy is to express the CoV-2 spike by cloning it into a gene and then making a virus that you see at the left, TNX 1800, that has the instructions to encode the CoV-2 spike protein. That spike protein is not expressed on the surface of the virus, but on the next slide, you'll see what we're hoping to do is to induce a kind of T cell immunity called Th1 immunity. There are two types of T cell immunity, Th1 and Th2. And as you can see on this schematic on the right, they're in a dynamic equilibrium, but it's almost never an equilibrium, an even equilibrium. So I show it as kind of a seesaw, where if you have a strong Th1 response, the Th2 response is turned off. And if you have a strong Th2 response, the Th1 response is turned off. And our hypothesis is that to protect against COVID, you need to induce a Th1 response. And that's pretty much the same as talking about a cellular immune response. Now, there certainly are antibodies produced in a Th1 response, but it's got a strong cellular component where T cells are turned on. And just last week on Friday, there were two papers showing T cell responses to the spike protein from one group and in all the patients, and the other group in the majority of cases. So as the clinical information comes out, I think more and more attention will be on T cell responses. So the next slide shows that 1800, our potential vaccine, is designed to infect skin cells after administration, and it instructs the skin cells to express the spike protein on their surface. It's a live replicating virus, which has the advantage of being a very strong stimulus to the immune system. And so it does not, for example, require any additional adjuvant. But also, because it's a live vaccine, the dose that we need is quite low, so that it would be possible to make enough for the United States, or even more than the United States, with a relatively small number of manufacturing runs. And this is exemplified by the fact that this is quite similar to the vaccinia virus that was used historically to protect against smallpox. The next slide. And similar to the traditional smallpox vaccines, ours is administered by scarification, which is a classic bifurcated needle, several pokes into the arm, and it results in a take or a cutaneous reaction that's shown on the right. And the cells shown there are a magnification of all the things going on within the major cutaneous reaction. But that's how the immune cells in the body, T cells and B cells, get activated to recognize the spike protein. Next slide. One of the, as I mentioned, the relationship of horsepox to the traditional vaccinia proteins, and there really are some differences that point us to believe that horsepox is really a primordial virus relative to the vaccinia. So it's related to the primordial or progenitor virus of the vaccinia. And the vaccinia have lost these regions on the left and the right side that are near what's called the inverted terminal repeats. You can see graphically here. At the top is horsepox in dark blue, and beneath it are a lot of vaccinia strains. And these regions control host cell response, and these genes that are encoded there interact with host cell proteins, which means that the horsepox has a less virulent phenotype than vaccinia, which we think will be amenable to more widespread use. Next slide. So we're collaborating with Southern Research, which is a well-known firm to do this, and they're working with us on both developing the product and also testing it in small and large animals. So what you can look for from us in the near future is our manufacturing plans and animal data, which we hope will be coming this year.
So what's the time frame for this?
We expect to have small and large non-human primate data in the fourth quarter of this year.
Okay.
Yay.
And then where does it go from there?
Well, we also hope in about the same time frame to have GMP material for human studies.
Great. Well, Eric hasn't joined us yet, so let's go to a couple of questions. Alex, this is for you.
Yeah.
Can you kindly explain what kind of nanoparticle encapsulation has been used by Moderna in developing their mRNA vaccine?
Hold on. Let me get to it. And yeah, this is a little bit more of the weeds. They use a novel lipid nanoparticle encapsulated vaccine encoding for a prefusion stabilized form of the spike protein. And beyond that, and not as up on the details of that and the formulation.
Okay. Did Eric just join us? I thought I saw Eric.
Yes, I'm here.
Oh, good. Okay.
Yeah, I was here a little bit earlier as well. So
I'm
sorry it didn't show up.
I didn't see you. Okay. So Eric, tell us about what Mount Sinai is doing.
So should I share my screen, or how would you like me to proceed?
Just the Berman group will advance the screen. So just
Okay.
let them know when to go to the next slide. First
Yeah,
of all,
please.
tell us who you are.
Sure. So pleasure to be here today. So my name is Eric Liam. I'm the Chief Commercial Innovation Officer for Mount Sinai and the Icahn School of Medicine, as well as our graduate school. I'm also the president of Innovation Partners.
Great. Thanks for joining us.
No, thanks for having me today. So I only have a couple of slides, and so my presentation is going to be actually quite short. And I'm going to talk about just really one program, but one program that Mount Sinai pursued that is obviously connected with another. So it relates to the development of a immunoassay for detecting antibodies to SARS-CoV-2 in human beings, in individuals. Mount Sinai, at the beginning of the pandemic in New York City, initiated a convalescent plasma program. It was we started under an emergency use authorization and then moved to the national protocol. I think we're over 400 individuals that were treated. Simultaneously with launching the convalescent plasma program, Florian Kramer's lab, which studies serology of viruses, developed an ELISA, a two-step ELISA for monitoring antibody levels in individuals. That assay, we use that assay to identify individuals with higher levels of antibodies against the virus for donation for the convalescent plasma program, and we've performed this assay well over 30,000 patient samples now. The assay itself, as I pointed out, is two ELISAs that are run in serial. The initial ELISA is against the receptor binding domain of the spike protein, and when we get a positive result in that assay, a second ELISA is run against the full-length spike protein, and the second ELISA is a titration, so providing us a titer of the level of antibodies that an individual has. I think I have performance data on the next slide. Yeah, so I have some performance data at the end there. In regard to distinguishing characteristics, as I pointed out, this was really developed to support our convalescent plasma program. But as we were doing that, and we received emergency use authorization from the New York State Department of Health, it became clear that this could be more broadly applied. We also have received an emergency use authorization for the FDA and are continuing to advance the capabilities of the test. This is an ELISA. We recognize that it's not high throughput, and actually specifically have gone forward with developing this assay and initial steps to make it available much more broadly to clinical laboratories, because it can be performed by really any licensed clinical laboratory. We recognize that it has potential uses in vaccine development and immunotherapeutics as well. The features that we're working on now are shifting it toward a semi-quantitative assay. We're working on that. And we've also been doing an extensive amount of work related to understanding what levels of antibody are required, at least in vitro assays in laboratories, for neutralization of the virus. Again, as I alluded to a little earlier, the statistics in terms of this, or the performance in terms of this assay, we have a positive predictive value of 100% with an NPV of 99.6. Now, I should point out that those were actually calculated by the NCI in their practice of the, or analysis of the assay. The specificity is 100%, and the specificity currently is 94%. One of the things I should point out regarding specificity is that because we started doing assays very early in the crisis in New York City, we were assaying individuals that were just a few days out from the onset of symptoms. We note that for other assays, they are indicated for use 14 days after, for example, 14 days after a positive PCR, or just a nucleic acid test. And well, what we see in all of the serology studies that we've done is that by 14 to 18 days out, you're seeing significant responses in the vast majority of individuals. So when you're testing at those later stages, the amount of antibody, the immune response is much higher. So again, we're working very hard on advancing the features of this assay, and are also utilizing this in various other programs that we have, considering that the assay recognizes the spike protein. I don't think I need to go into the details since it was covered earlier that spike is obviously required for entry into cells and believed to be blocking it, but is believed to be protective. So that's all I was going to comment on today, and I'm happy to answer any questions that you have.
So Eric, you want to make this broadly available to others. How will you do that? Will it be through some kind of a universal license or a special license?
We've made the technology available broadly in the academic community, and have also made it available through licensing with commercial entities. We're also partnering directly for the development of kits that will be available to any licensed clinical laboratory for purchase, assuming that translation of the assay goes according to plan. Yeah, those will be available in the next months.
Okay, great, thank you. Let's look at a couple of questions from the audience now, and this was actually on my mind as well. Assuming more than one vaccine could prove to be efficacious, how would these be rolled out? And in cadencing the recipients, how would it be determined who will get the vaccine? My question was more along the lines of, if more than one vaccine is developed, how do you choose which one to take?
I think, you know, we talk a lot about precision medicine or personalized medicine. These vaccines are going to have to distinguish themselves in trials by super serving or being most effective on a subpopulation of patients. And I think on that basis, we can have several candidates. Now, once we have them, there's going to be a couple of issues. How are we going to get this into the hands of patients? There'll be payment issues with insurance, but also how, pricing of some of these therapies, and what, if any, efforts will emerge worldwide to try to either pool intellectual property or allow for multiple users of the technology. Might we get into some sort of a license for use of these? Now, there are efforts toward that, but they don't seem to have buy-in from biopharma companies. For example, last week, the World Health Organization endorsed the idea of a voluntary pool to collect patent rights, and test data, and other info that could be shared by companies. But again, those were nonprofits and other groups, stakeholders, the same, but not the companies themselves. And not to mention is the ongoing issue of cooperating with the World Health Organization. You've seen the United States threaten to pull out. The U.S. joined with a few other countries in staying away from even a European effort toward that. That was the Coronavirus Global Response Pledging event by the European Commission. So the subgroups will, they'll be distinguished, but there's other issues beyond just before we can get this into the hands of the millions and millions of patients we need to get this to.
So David, here's one for you. What are, if any, the largest analytical challenges facing COVID-19 treatment and vaccine development?
Analytic challenges. Well, I think probably the first one is we have to make sure that the tests that we're using, whether it's to determine infectivity, some variation of a PCR, or whether it's to exclude patients who have already been infected and have seroconverted on their own, are accurate. We have a team at Regeneron that's been looking at the various different tests that are out there because this is a critical question if we're going to conduct studies, whether it's with our cocktail or a vaccine. And there's a lot of variability into the specificity and sensitivity of the tests that are currently being used, sort of in the tents in parking lots of hospitals. And in this has to get solved. If the tests are inaccurate, then we're going to have a problem when we start trying to evaluate efficacy for any of these potential therapeutics.
Okay, and Seth, here's one for you. Can the spike protein of SARS-CoV-2 be ligated on the horsepox virus, be expressed on the horsepox virus surface just like it is expressed on SARS-CoV-2 surface in order to induce Th1 immune response?
Thanks, that's a good question. The way horsepox works is that the gene would be inside it, but there won't be CoV-2 on the surface, CoV-2 spike. But once it infects the skin cells in the arm at the site of the injection, then those cells express spike, and they express it in a way quite a bit like the virus does. So it's not really necessary that the virus itself express it, but the cells that it infects. And this is different. Ours is a live replicated virus. And that is different from others. For example, the J&J or the Oxford viruses are non-replicating. And I think that that's a difference. But so the virus that will come out will not express it, but the cells that it will then infect in the arm will express it.
Great, thank you. Eric, kind of specifically, on what panel of cross-reactions was 100% specificity based? And then more broadly, what benefits has Mount Sinai seen from partnering with Sorrento Therapeutics?
So the way that the assay was validated is, in fact, let me just start over. It was very important for obviously having positive controls and negative controls to validate the assay. Mount Sinai has a large body of banked plasma that actually dates from before COVID-19. We have samples that are validated for containing antibodies to HIV, other coronaviruses, flu, etc. So by using both knowns and just general plasma samples from population prior to COVID-19, we were able to, with many samples, demonstrate that we had no cross-reactivity. So again, both specific as well as general use of plasma predating the outbreak. In terms of positive controls, we have samples from individuals early, mid, late stage, post recovery. Mount Sinai, I believe, is approaching 6,000. I've heard over 6,000 patients that have returned home from being treated at Mount Sinai. So,
congratulations.
Thank you. Yes, it's. We're pleased that the numbers are trending in the right direction, obviously. I mean, not to state the obvious, but our ability to actually validate the assay is quite significant. We also were able to share samples with other entities, the government, the state, for use in their work as well. And that's been, that was announced in a press release by the government about 2 weeks ago. In terms of our relationship with Sorrento, that's obviously a different relationship that seeks to develop therapeutic antibodies for prophylactic or therapeutic treatment of individuals in the future. Mount Sinai obviously has a very large population of individuals that we know have high titers for antibodies as well. And, similar to the convalescent plasma program, we're pursuing additional programs that would support production of products in that space.
Great. So we're just about out of time. So I want to wrap up with, one more general question, and that is timing important here? If one company comes up with a vaccine, what happens to everybody else's trials? And who gets the vaccine first, both globally and nationally? And how do those decisions get made? And I'd like each of you just to jump in here and give a thought.
Sure. Well, I'll start. I think that there won't be a one-size-fits-all model for vaccines. For example, our vaccine can only safely be used on healthy, immunocompetent, non-pregnant adults. So, that's the majority of people, but not everyone, and there are obviously important groups of people that won't be addressed. So I think to some extent, that's the beginning of one of the selections. But clearly, I think it's so exciting to see the rollout of some of these vaccines, and we'll be getting data real time, and I'm sure people will be adapting to it.
I think the decisions are going to come government by government, country by country. There are global bodies like the World Health, for example, like the European Union, but I think that there's a lot of pressure. I mentioned earlier this whole the nation's efforts to develop first dibs. I believe, no, that's not it. But so I see that I don't see any one body being able to say, okay, everybody gets it. There isn't. It's just these things are regulated country to country. So I think ultimately access will be determined. There'll be approvals needed in the different places. So that's going to be a challenge because poorer parts of the world may not see the vaccine or drugs that are being developed as early unless other groups make an effort to bring them to distressed parts of the world.
So David, you've seen this issue with using your platform with other infectious diseases like Ebola. If another vaccine is developed, what happens to your efforts?
Well, I think we've always looked at the antibody cocktail as a bridge to a long-standing vaccine. I mean, in the long run, a traditional vaccine is going to be simpler and more economical to deploy in an incredibly broad fashion than an antibody. But there's a bridge that needs to be built between being able to potentially show that a therapeutic, that the antibody works in a prophylaxis setting, and the vaccine trials. But in addition, an antibody may be useful in a therapeutic sense if the patient already has the disease now, or in a future state if the vaccine is partially effective and not completely effective. But to be perfectly honest, I don't think we really care. The question of whether or not there's going to be more than one potential treatment here, that's a good thing. We have to work collectively to weed out what's going to work and what's not going to work, and concentrate our efforts in a handful of places so that we're not all stumbling completely over one another and no one gets an answer. But we want to beat this. This isn't a drug that at least we're not developing principally for traditional commercial purposes. We're developing it to try and cure the world. And if a better mousetrap gets built, it just takes an extra year. There was huge value in doing this because I don't know about you guys, but I'd prefer not to keep the world closed down for a year waiting for the vaccine trials. So like I said, the typical kind of commercial mindset, for a for-profit biopharmaceutical company, I don't know about everybody else, but we've kind of thrown that out the window. We're doing this for something bigger.
Yeah.
Yeah, Nancy, I was going to say that there's good examples of having multiple vaccines available, right? Even with flu, we have multiple vaccines, and there's room for them. And there's really different populations may use them, right? Different delivery modes, for example. And then there's also individuals that, for whatever health reason, legitimately may have reactions. Individuals that are, for example, allergic to eggs are not able to take certain vaccines, right? Versus others that are other vaccines that are made different ways. And so, I think that those issues come into play here. And I really like what David said about bridging, because I think if you go from convalescent plasma to, kind of a hyperimmune globulin to a clonal immune therapeutic, all of these are steps in the right direction to provide us tools to combat the virus. Well, the ultimate tool is a vaccine, one or more, that elicits, in theory, a long-term sustainable immune response in individuals. But that's another thing that we're going to have to monitor, because, our understanding, or my understanding from from our team of researchers at Mount Sinai, is that in seasonal coronaviruses that tend to cause more of the seasonal colds, that you see 18 months of immunity, something like that. And so, that's a big question. What's the durability of immunity that we're going to get? And I also think it's really, David made another point, which is really critical. We've got to have good testing. And the whole purpose of what we're trying to do is to take the test that we developed that we believe is extremely good, and to make it available to, broadly to the community so that they can use it, both now in terms of understanding whether there's an individual has an immune response, but potentially in the future as well to look at, well, what's the durability of that response?
Right. Right. Well, we're going to have to close now, but we did have one closing question that I just want to get in here. In terms of deciding who gets the vaccine, would age and vulnerability set the priority?
I could see governments making that decision based on perceived vulnerability or age. They could, but that's yet to be, it's a framework that's yet to be established. And I think that's going to have to be established in the next year or two, because definitely given the pace of drug and vaccine development, we're going to get things that pass through clinical trials that will show safety and effectiveness and that will be approved. But without figuring out who gets it first, then what good is it if it doesn't get into the hands of as many patients as needed?
Eric, you actually treat patients, so how would Mount Sinai deal with a question like this?
I don't know if I want to answer this question. This is a complex question. Look, at Mount Sinai, our goal is to serve all of our patients. We would hope that there are solutions for all of our patients. Different types of vaccines, different solutions. Again, as I commented on immune therapies, I think all of these are part of the solution. I agree that if those kind of decisions, Nancy, are going to have to be made, that's probably more of a governmental level. We're going to obviously, to the degree that there are governmental directives, we would follow those. But we're going to try to serve all of our patients and serve the needs that they have, and use every tool that's available to ensure that we can, move beyond this situation.
Well, that's a wonderful note to end on. So I thank all of you for coming and participating and educating our audience this afternoon about the vaccines and treatments in development for COVID-19. Remember, we will be posting this on the website, so if any of you want to go back and listen to portions of it, you'll be able to listen, and the presentations will be there also. Thanks, everyone. Have a wonderful day.
Thank you. Take care.
Thank you. Take care.
Well, look into it.